Hi, hello, I'm Shishra Joy, second MSc Biology student in St. Mary's College, Thrissur. Now we are discussing about the topic determination of the primary structure of DNA. We are only discussing non-spectrometric method, DNA sequencing method, and Edman degradation. In non-spectrometric method, column chromatography, NSDS space. In DNA sequencing method, Sanger's method, Maxim and Gilbert method, hybridization, Palnaesan method, slab gel sequencing, and capillary gel electrophoresis. Then third one, Edman degradation. So first one is the non-spectrometric method. In this method, the column chromatography. In this type of chromatography, stationary phase setup is placed inside a tube. Stationary phase may be silica, alumina or cellulose. There are two methods, that is wet method and dry method. In wet method, stationary phase and mobile phase mixed and form slurry, but in dry method, stationary phase is powdered. In the bottom of the tube is filled with cotton or glass wool. Based on affinity of sample and stationary phase, it gets separated. Next one is the SDS space. It's a type of gel electrophoresis. This is a technique used for separation of protein based on the molecular weight. It is a technique widely used in forensic, genetics, biotechnology, and molecular biology to separate the protein molecule based on their electrophoretic mobility. Protein is a large complex molecule. First, its bond break down and form linear structure by beta mercaptoethanol. So we got a linear structure of the protein. Then we want to introduce a negative charge. With help of SDS, we introduce negative charge all over the linear DNA. We took the polyacrylamide. It has its own particularity. It's introduced into the gel casting plate and make well by a comb and add distilled water above the gel. This is the procedure of SDS page. We load the protein sample and marker in this well. Electricity is applied. So DNA is moved based on the charge and mass, and we got separated. The next one is Sanger's method. It is also called dideoxy sequencing. Deoxyribonucleic acid, which means oxygen is absent in the second position. So what about the dideoxy pentose sugar? In dideoxy pentose sugar, oxygen is absent in second position as well as the third position. Just think, new nucleotide is added to the 3' end of the ribonucleic acid. So in dideoxy pentose sugar, oxygen is absent in third position. So it's unable to make a bond between them. So it can form the nucleotide in the. This property is used in the Sanger's method. Here dideoxy pentose sugar is used for fragmentation of DNA. So this method is also called dideoxy sequencing. This is the procedure of Sanger's method. First we take the 4 test tube. In the first test tube, we add single stranded DNA with unknown sequence serve as a template, DNA polymerase, DDATP, DCTP, DTP and DGTP. The second test tube, we add the single stranded DNA, DNA polymerase, DATP, DDCTP, DTTP and GTP. The third test tube, we added single stranded DNA, DNA polymerase, DATP, DCTP, DDTTP and DGTP. The fourth test tube, we added single stranded DNA, DNA polymerase, DATP, DCTP, DDTP and DDGTP. We take first test tube. Here single stranded DNA with unknown sequence. We also put dideoxy ATP into it. So this dideoxy ATP bind to the thymine. At this position, adding of the nucleotide is completely stopped because of absence of oxygen in the 3' end. So this process takes place in the rest of 3 test tube, and it is undergo gel electrophoresis followed by autoradiography. Sequence can be read from the band's autoradiograph and original templates sequence reduced. Longest fragment ends with ADDZ, so Z must be the last base in the sequence. So this undergo gel electrophoresis and we can detect the sequence of the DNA. Next one is the Maxim Gilbert method. Based on nucleobase specific partial chemical modification of DNA and subsequent cleavage of DNA backbone at a site adjacent to the modified nucleotide. By chemical method, sample DNA is converted to single-stranded DNA. Then added P32 as 5-phosphate. 4 reactions may happen, that is A reaction, G reaction, C reaction and T reaction. So this is the procedure. The sample DNA, it's undergo single stranded DNA, then added P32 as a 5-phosphate, cleavage at specific nucleotide. First we take the G reaction. In the first case, in the G reaction, guanine is cut from where guanine is. It is also happening A reaction, T reaction and C reaction. So DNA get fragmented at specific site, then it undergo electrophoresis, then it undergo the radioautography, so we can find out the sequence of the DNA. The next method is the hybridization. Double-stranded DNA is converted to single-stranded DNA by increasing temperature. Single-stranded DNA is added to nitrocellulose. Then probe is introduced in it. Probe is paired with the complement base pair of DNA present in it. Next one is Pal-Nielsen's method. It is also called Biosequencing. So Biosequencing is a DNA sequencing technique based on sequencing by synthesis enabling rapid real-time sequence determination. So here double-stranded DNA is converted into single-stranded DNA. This single-stranded DNA, primer and DNA polymerase are put into a sequencing well. So we take this situation, here we had single-stranded DNA. Then nucleotide is added. When the time of each nucleotide added, 2-phosphate group is released. So this is called the pyrophosphate method. So in the time of nucleotide added, 2-phosphate group is released. That's why it's called pyrophosphate sequencing. This pyrophosphate with APC, it forms ATP with the help of ATP sulfurylase. So this pyrophosphate is converted to the ATP. In that well, we also added luciferin and luciferase. This ATP convert luciferin into oxyluciferin with the help of luciferase. When this luciferin is converted to the oxyluciferin, light is emitted. This light is detected by a specific detector. By this method, we can find out the DNA sequence. The next one is slab cell electrophoresis. It's also known as SDS phase. SDS phase we discussed before. The next one is the capillary gel electrophoresis. DNA separated on the basis of charge to mass ratio. Applied 1000 voltage. A positive and negative electrode are placed in a solution contains ions. Then when a voltage is applied across the electrode, solute ions of different charge, that is anions and cations, will move through the solution towards the electrode of opposite charges. Capillary electrophoresis then is a technique performing electrophoresis in buffer-filled narrow bore capillaries, normally from 25 to 100 mm in internal diameter. So this is the flow chart of capillary cell electrophoresis. The last one is the Edmond degradation. It is also called phenyl isothiocyanate sequential process. So Protein is bind with PITC, phenyl isothiocyanate. When it get attached, only one nucleotide in the N terminal will persist and other is removed. So this complex is called phenyl thiocarbamyl tripeptide derivative, and this is converted to phenyl thiohydination derivatives, that is the PTH. PTH amino acid undergo the analysis by HPLC. We can find out the DNA sequence.